This is a podcast which looks at DNA in more detail, and it really is quite an incredible molecule. It has been around for a huge amount of time, and it is a genetic material which is passed on from generation to generation, which gives the blueprint of the characteristics of every organism that there is. And I want to start off by really just putting it into context, and that is that if you think about DNA, it is really just a code. And we are going to talk about adenine, guanine, cytosine, thymine, which are really strange words. But I'd like us just to realise that, in fact, we deal with codes the whole time. And if we were to say, for example, the letter C, and it doesn't really mean anything. The letter A doesn't really mean anything to us. The letter R doesn't really mean anything. But if we actually put those together with the code that we use every day, which is the alphabet, we suddenly get a very badly drawn object with a steering wheel, 4 wheels, lights, windscreen. Amazing. So just by putting 3 letters together, we are able to translate that into a physical object which has so many attributes that we ourselves understand. And this is exactly the same for DNA. But instead of it being our alphabet of 26 letters, we just have the letters A, T, G, and C. And usually, instead of it being just 3 letters, like our alphabet, which can make the image of a car, we usually have, say, 150,000 letters, which would form the equivalent of an object, like a car, which is, for us, a protein. So in DNA language, instead of forming an object with 4 wheels, steering wheel, lights, shiny body, we might go to a protein of thousands of amino acids, which looks like that. And to our cells, that is every bit as specific and recognisable as a car, but it's used a different language, a different code. Instead of our alphabet, it's just used 4 letters. So it's not that alien to us at all, in fact. What is alien is that if you were to put 150,000 A, T, G and C's in a certain order, it would mean nothing to us. You can't even pronounce that word, but the truth is, for our cells, DNA is a code which can be translated into proteins, and that's hugely important and incredible. So, moving on, we need to look at DNA in a little more detail. And it's made up of nucleotides. And we can, in fact, look at a more recognisable DNA double strand. And we would know that as a diagrammatic format, DNA takes the form of that. We have 2 strands, 1, 2. And they form like this. We can see here, and hopefully our biological knowledge so far hasn't really introduced anything too difficult here from translating this diagrammatic double strand to this one in the centre. We have bases, sugars, and phosphates, which line up, and we have 1 nucleotide there, bound to another nucleotide there, bound to another nucleotide there. And what we need to know is the basic structure of a nucleotide, and we need to know how they bond together to form a polynucleotide, poly meaning many, and the way that those polynucleotides interact with each other in strands 1 and 2. You can see that the structure is the same as well, in that this strand has nucleotide, nucleotide, and nucleotide. So that is how DNA is bonded. Let's go back and let's just look at that nucleotide on its own. So this is our basic nucleotide structure, and it's easy as 1, 2, 3. 1, the phosphate group. 2, a 5-carbon pentose sugar, 5 meaning pent or pent meaning 5, and 3, a nitrogenous base. Now, we were looking at DNA, we've actually come back to a more general position here with a nucleotide, because both DNA and RNA are made of nucleotides. There's a couple of differences, though, that exist between DNA and RNA. The first is that DNA, the sugar, the ribose sugar, is a deoxyribose. That means it's without one of the oxygens that ribose has. So for RNA, the sugar is ribose, and for DNA is deoxyribose. In DNA, this nitrogenous base can be adenine, thymine, guanine, and cytosine, whereas in RNA, the nitrogenous base can be adenine, uracil, guanine, and cytosine. The last difference, which I'll just mention briefly, is the fact that, as we've already mentioned, DNA forms double strands and RNA actually only forms single strands. So there is our basic nucleotide. So how do they bond together? Well, they form, keyword, phosphodiester bonds. And each nucleotide is an identical monomer. And this is a process of polymerization to form a polynucleotide. And it is, in fact, very simple. Here we have 2 individual nucleotides. They are identical. We've got a phosphate bound to the 5th carbon and a base bound to the 1st carbon. And in fact, if we number these carbons, we've got 1, 2, and then you can see the 3 is already written in, 4 is there, and then the 5 is there. And they're going to bond together by this phosphodiester bond. And what happens is that the phosphate group binds to the 3rd carbon, and this is the phosphodiester bond. Easy. And so, if we were going to extend this, because the 3rd carbon bonds to the phosphate group of another nucleotide, we would come down here to add a nucleotide, and we'd form a phosphodiester bond, and it would be as easy as that. Phosphodiester bond between the phosphate group adding on to the 3rd carbon. I'd like to stress something here for later on, and that is that the phosphate group of a new nucleotide will always form a phosphodiester bond with the 3rd carbon. The 3rd carbon. Okay, that means that a chain of polynucleotides where a nucleotide is being added will always be added in a direction where it starts off at 5 and then 1 is added to the 3rd, then 1 is added to the 3rd, 1 is added to the 3rd, 1 is added to the 3rd, 1 is added to the 3rd. And what I mean by that is that we have one here, and we have one here, and then we'll add one at the third carbon below, and the third carbon below. And so can you see that actually the addition is travelling in that direction? This is really important because DNA replication or messenger RNA transcription that we're going to learn always travels in a 5' to 3' direction. There is always a replication where nucleotides are added in this direction, which we need to remember. So, that is how polynucleotides form from individual nucleotides. So, if we go back to our diagram here, and I get rid of some of this annotation that I did earlier, we are now left with DNA and the exact structure of it. We have the nucleotide here, which has got a phosphodiester bond here, which bonds it to another nucleotide. Next thing I want to mention is the bases. Now there are two types of bases. There are the purines and the pyrimidines. And each of these bases is either a purine or a pyrimidine. They form hydrogen bonds between each other. And that is the mechanism by which two strands are held together. So the next area that we need to know about are which are purines and which are pyrimidines, and how many hydrogen bonds form between them. Well, the purines we can see on this diagram are adenine and guanine. So these are the purines. The pyrimidines are cytosine and thymine. And an adenine and thymine bond is 2 hydrogen bonds, and a guanine and cytosine is 3. There's 3, and there's 2. And this is important because we need to know that the hydrogen bonds that keep these strands together are the bonds that are broken when an enzyme called helicase unwinds the strand. So this is our normal DNA, and as they unwind, helicase is going to break these hydrogen bonds and therefore unwind the strands.